All right, population genetics and Hardy-Weinberg equilibrium. So population genetics, first we have to define what a population is. And that's a group of individuals of the same species that live in the same area and they interbreed to produce fertile offspring, remember? So the gene pool is a population's genetic makeup. It consists of all the copies of every type of allele, and if there's only one allele present for this particular gene or locus, the loci of where it is on a chromosome, if this is in a population and it is fixed, then many fixed alleles equals less genetic diversity. So more importantly, I always remember gene pool by an image. This is my gene pool of mice. If I have two black mice that are homozygous dominant for hair color, big B, big B, big B, big B, then each of those alleles go into a pool. The gray mouse, which is little b, homozygous recessive, is donating, I think about it, two recessive alleles to the pool. And then if we have a trait where there's codominance displayed, which means that each allele is going to get credit or be expressed, creating a third phenotype or a different expressive phenotype, like my, let's call it tan mouse right here, which he is heterozygous. He has a big B and a little b, and therefore both of those are expressed, giving him a different coat color. Then those go into the pool. And the accumulation of all the alleles that are available for making offspring are our pool. And this is important to view this way or think about it when it comes to the math and how we calculate this. In population genetics, we can refer to the allele frequency in populations that's going to change over time. So remember that populations are evolving, not individuals. So in images or calculations where population genetics is discussed, we are referring to a population, most oftentimes that is isolated. Microevolution, that is small scale genetics, and those are small changes in a population. Evolution is driven by these random occurrences. They can be mutations, genetic drift, migration, which is also called gene flow, and then, of course, natural selection. You may have remember us discussing mutations before, but let's just review. Okay, so a mutation in your own words, like mutation is basically a genetic variation, and it can happen in the form of a new allele. Natural selection it acts on a variety of different things, mostly phenotypes that we see. And mutation rates, they tend to be slow in plants and then in animals, and then of course very fast in prokaryotes, just due to the generation time and then being able to be reproduce generations more quickly. So it's important to note that mutations, they can be harmful, neutral, or beneficial. No one said that this is in the up direction when we refer to mutations, but most mutations are in the neutral to harmful range. Not all mutations are going to lead to evolution or success in helping the species. Another mechanism for population genetics is genetic drift. This is a chance event that is caused by a change in allele frequency from the generation to the next. Most significant to these really small populations where a chance event can just completely devastate and rapidly change the genetic information in a smaller population. But this can also lead to complete genetic diversity that's lost, and so it can also make a harmful allele become fixed in a population, but oftentimes it doesn't really produce an adaptation. These are things like the bottleneck effect or a founder effect. But it's some sort of random chance that has occurred and has devastated or removed a significant amount of the population. Bottleneck is when a large population is drastically reduced. This is like a non-selective disaster. It could be like a fire or a famine or a hurricane, something like that. Like some alleles may become overrepresented or underrepresented in this case, or a population could completely lose a set of genetic patterns completely. But this is when there's a very, very small amount of survivors from a specific population. In the founder effect, this is when few individuals become isolated from a large population. And this new established population, which is relatively small, are going to start a whole new gene pool that's available. This does get our create a genetic loss. There's a lot of loss in diversity because only a few individuals were selected to be part of a new population. And sometimes humans, they can definitely impact this, or it could just be a gust of wind that blows a couple of birds all the way out to a new volcanic island. When we transfer these alleles in or out of a group of a population, usually due to some fertile individuals or just gametes, we call that gene flow. Alleles, they can be transferred between a population, just by being moved physically, like by being blown. If pollen is always blown into the wind and then it finds like a new location. Okay, so we're just gonna recap and review and give some scenarios and some examples. We're gonna examine each scenario and determine if it's an example of the bottleneck effect, the founder effect, or genetic drift, and you want to be able to justify each of response with an explanation. So let's try some examples here. Scenario 1, there is a black robin and it's a small bird. It's native to Chatham Island, which is part of New Zealand. And before the 19th century, much of their habitat was converted to farmland. So due to the loss of their habitat and the introduction of non-native predators, more like cats, the population declined to its lowest point. Only 5 individuals. And since then, conservation efforts have been brought about in this population and area to bring the number of these species back. And now we're up to an estimated of 230 individuals. Can you identify this mechanism? It is the bottleneck effect, and this is also due to human activity. Scenario number 2. In the 1720s, an Amish population settled into Pennsylvania. A small number of the early settlers were from Germany, and they carried an unusual number of mutations, one of which included polydactyly. The trait for polydactyly is now much more common in the Amish population than it is in the general population. So can you tell me what mechanism this would be? It's the founder effect. So few individuals from a larger population came over to another area and they were isolated, and so now that trait has become more evident in the population. Scenario number 3. This recurrent flash floods, they have caused this mass mortalities in the marble trout population. Scientists have analyzed that the genetic makeup of the remaining marble trout population, and they have found that the genetic diversity has decreased significantly, which in this scenario we can assume that this is due to the flash floods. Here again we have the bottleneck effect, where something has caused this a disaster, has taken a population, and then high mortality basically removed a lot of the genetic variation in a population, and we were left to a very small non-diverse genetic group. Due to an increase in wind in a local area, pollen from one population of flowers has traveled further than usual and pollinated another population of flowers. The offspring from these flowers now have traits of each population. Can you give me an idea what mechanism this is? It's an example of gene flow. Like literally gene flow with the wind. So now that you're familiar with natural selection's mechanisms, we're going to take a closer look at how it can actually affect a population. All right, reproductive success is measured by relative fitness. This is the number of surviving offspring that an individual produces compared to the number left by others in the population. So the effects of natural selection can be measured by examining the changes in the mean average of the phenotypes. There are 3 modes of natural selection. So they are directional, stabilizing, and disruptive. An AP biology student can be asked about a mode for natural selection in a couple ways. They could give you an example and have you determine the mode of selection. They could give you graphs and have you illustrate on top of a graph what would happen before or after selection. So, and that's just a couple of different ways. You can be asked this a lot of different ways. So it's important that you understand what's happening here. In the modes of natural selection, we have in blue, that is after the selection process. So in red, we have before the selection process. Look at all three of the modes of natural selection, and these are our populations before anything is acted upon them. Okay? If you look at the red, it's almost identical in all three types of selective process. So really what this is how a mode of natural selection can act on a population that has a normal, just shape, rounded curve, if you will. If something is directional, then it's going to select towards one extreme of the phenotype, all right? And usually that you'll see that more illustrated towards the right or towards the left, but it's just one. So our after selection on directional is moving towards one side of the population. Stabilizing selection will select more towards the average or the mean against all types of extreme phenotypes. So we're going to have a higher amount of the population kind of smushed into the center part there, stabilizing that. And then disruptive selection is going to select against the mean. So both phenotypic extremes have the highest relative fitness, and so it's disrupting the normal mean and selecting against extremes. Sexual selection is a type of natural selection that explains why many species have unique or showy traits. And most often this happens in males. Males are often, they have these useless structures like the colorful feathers of a peacock. This is just simply because the females, they like it. So they choose that trait when they're choosing a mate. It can produce traits that can be harmful to survival, though. So, those colorful feathers in male peacocks, they make them very easier to spot in predators. We've actually seen examples like this taking it all the way to extinction when females have selectively sexually selected for male phenotypes that have been detrimental to a population. Alrighty, Hardy-Weinberg. This is a model that uses, in order to assess whether or not a population is in natural selection or if there's other factors causing the evolution at a particular allele location site. So this determines what the genetic makeup of a population would be if it were not evolving. This is then compared to like actual data. So if there are no differences, then we consider the population not evolving. And if there are differences, then we may consider that the population may be evolving. Here we have again a mathematical calculation to back the science. Hardy-Weinberg principle states that the frequency of alleles and genotypes in a population will remain constant from generation to generation to generation, provided that only Mendelian segregation and recombination of alleles are at work. So remember, this is just a hypothetical situation where no evolution would take place. In real populations, the alleles and the genotype frequencies, they do change over time. So there must be 5 conditions in order for Hardy-Weinberg equilibrium to be met. There can be no random mutations, no random mating, no natural selection, no extreme large population size, and definitely no gene flow. Any of these conditions, if they are not met, then microevolution occurs, like a mutation or gene flow, genetic drift, selection, random mating, that type of thing. But in order for a population to be considered in Hardy-Weinberg equilibrium, none of those 5 things must be going on. So think about it for a minute. How each of those 5 Hardy-Weinberg conditions, how if they're not going to be met, then how are they leading to evolution? There are 2 mathematical expressions that we use in order to determine Hardy-Weinberg equilibrium. They are P plus Q equals 1. So P is going to be the frequency of the dominant allele in a given population, and Q is the frequency of the recessive allele in a given population. And because each individuals have either one or both of those, then if we add those two up, we should get 1. The second formula is going to also equal 1, but we can be given the percentages of the homozygous dominant individuals, the percentages of the heterozygous individuals, and then the percentages of the homozygous recessive individuals. So the second formula takes into account this heterozygous individuals they're visible within a population. So let's just make sure you understand, if you are told that 20% of the population is homozygous recessive, what variable are we referring to for the Hardy-Weinberg equations, P or Q? It's actually referring to Q squared. Let's try this again. If you are told that the frequency of a dominant allele in a population is 75%, what variable is referring to in the Hardy-Weinberg equilibrium? That's referring to P. So in this example, we were given a frequency. We're only talking dominant recessive alleles. We know the frequency of that dominant allele is 75%, so that is P. But in the first example, we're talking about a percentage of a population. So we're going to have to use the second equation to determine where that 20% thing is supposed to go. When you're trying to solve these Hardy-Weinberg equilibrium problems, you want to just first of all start with which formula that you can start with. If the problem gives you allele frequencies, it's usually referring to that P and Q almost all of the time. If it gives you more information about individual organisms or populations or percentages or even mentions the heterozygous comment, then it's referring to that P squared, 2PQ, and Q squared formula. But most time you will use both of the formulas to complete the problem. And usually you're given Q, and then you'll need to basically find P, and then also apply that to the other problems in the second equation. So just some tips on how I solve for them. I always write down both equations at the top, and then I identify the information that's given. Like, am I given the allele frequency in that population? Regardless of what the problem asks, I always solve for both P and Q first, because I know that's going to allow me to answer the question or part of the question or some of the question. And you can use a calculator while doing this. Always use a calculator to square and square root functions, because you don't want to just make a mistake and then your problems don't equal up to 1 because of you trying to remember your square root functions. The most common mistake that students make is assuming that they're going to know the answer because they have, they can square it and square root everything in their head. The next is a set of 4 practice problems that I'm going to give you a moment to do the answer to and then check your answer. Were you able to get the allele frequency of little a is 60%, while the frequency of big A is 40%? Let's try this again. Practice problem number 2.
Okay, so sorry about earlier.
And the answer to number 3, 55%. Try the last example here. And the answer is that the big B allele frequency is 35% and little b is 65%. So let's review some content here. Like, what is the difference between genotype frequencies and allele frequencies in a population? So genotype frequencies, that's going to refer to like all the alleles combined. And allele frequency is going to refer to the allele's relative distribution, like how common the allele is. Hardy-Weinberg is often referred to as the null model of evolution. And what this means is that the null model of evolution is one where no evolution is really occurring, which is assumed what Hardy-Weinberg needs in order for all of its conditions to be met. More often than not, there's genetic diversity in a population. Um, the better it can respond to the environment. So, genetic variation is considered an advantage, but more likely for individuals to withstand sort of changes. Like species with this low genetic diversity, they're at risk for decline or extinction. So, just as an example, due to poaching and poisoning, the California condor was reduced to 27 individuals, and this drastically lowered the gene pool. And even through this restoration effort, we've seen this increase in numbers, but there's a lot of genetic diversity that has been lost. And we've seen this in lots and lots of populations that humans have tried to conserve and have come back. The original population that was there before humans impacted it was usually very genetically diverse and had a lot of diverse alleles. But once we remove a huge amount of that population for whatever reason, even if we stop the influence and they're allowed to reproduce again, we still have lost advantageous mutations or just genetic diversity altogether. We see this a lot in larger animals too, like the cheetah.